My guest today is Professor Meret Aeppli, who is Assistant Professor and Head of the Soil Biogeochemistry Laboratory at EPFL in Switzerland. Her group aims to elucidate the fundamental principles and mechanisms of electron transfer reactions and their role in the biogeochemical cycling of carbon and metals in soil. Welcome, Meret.
Thank you, Gill.
Yeah, so this is a somewhat exotic but interesting topic. And so to have a common understanding, I thought so there was a textbook that you wrote in chapter 5 is entitled Redox Reactions in Soils. Redox, I understand, stands for Reduction Oxidation Reactions in Soils. And you say here in introduction, in reduction and oxidation reactions, atoms exchange electrons, causing changes in oxidation states and chemical species, and thus dramatically change the behavior of chemicals in soils. Redox reactions can be abiotic or biotically driven. And you say here in most natural systems, biological reactions have the greatest influence on redox processes. Yeah, so I was looking at some graphics, Merit. So the soil underneath our feet is a huge recycling system in itself, right? And there's so many things going on there. There is biological things, there is chemistry, pure chemistry, there is energy coming into it through plants, through photosynthesis. So this is presumably one of the most complex systems on Earth that we haven't really dug deep into, I think. So
yeah, we dug, but yeah.
Yeah, go ahead. Yeah, so set the context for redox reactions in soil.
So first I want to clarify the textbook that you're referring to. I didn't write that, but I read it, and I'm working in this realm of redox reactions in soils. Yeah. And as you said, soils are really complex systems because that's basically a soil is where the atmosphere meets the hydrosphere, the biosphere, and the geosphere. And so basically we can have a really large complexity of reactions that are happening in the soil, most of which, of course, we cannot just see by eyes because they're hidden underneath, basically in the soil. So redox, I'm particularly interested in redox reactions, which, as you said, involve the transfer of electrons between different elements. And these reactions are really fundamental to life on Earth because they allow organisms to gain energy for cellular processes. Mm-hmm. So maybe before we go into the soil, we can think about our own bodies. So we also use redox reactions to live. Mm-hmm. We eat food, and basically we extract electrons from the organic carbon that we consume as food, and we deposit them onto oxygen. Thereby we produce CO2 and we release energy to power our metabolism. And soil microorganisms basically do the same. Mm. They also eat organic carbons. Some organisms can also use different substances, but for now I think we're going to stay with organic carbon. They can extract electrons from that organic carbon, but in comparison to us humans, the microorganisms can use a wider variety of compounds that can accept these electrons. Mm. So our bodies only function with oxygen, but there are microorganisms that can use other compounds than oxygen, for example, nitrate or sulfate, or even minerals that contain redox-active metals. And so when you hear this list of like different compounds that the microorganisms can use, you can already like guess that these reactions are going to have an impact on many element cycles. So for now, I think let's stick with the carbon cycle because that's on everyone's mind with climate change, of course. So if we look at the carbon cycle in soil, it's really a substantial part of the global carbon cycle that we have. So the carbon comes into the soil basically from plants, as you said. So plants do photosynthesis to capture the energy of the sun into organic compounds or organic carbon. And these are also a series of redox reactions. Now, when this carbon enters the soil, the microorganisms can eat some of it, by oxidizing it back to CO2, for example, or other greenhouse gases, in, again, different redox reactions. And these effluxes that we see from this microbial respiration are really significant on the global scale. So they're estimated to be around 6 times larger than our anthropogenic emissions. So if you hear this number, you can imagine that if we have even very subtle changes in these redox processes in the soil, that can have really dramatic impacts on atmospheric CO2 levels and climate change. So there's really a need for us to understand what is driving these greenhouse gas effluxes and microbial respiration in the soil, and also how that could possibly change in a warmer climate across different soils.
So very simplistically, if I understand this correctly, Meret, so plants create carbon in some state. It's almost like an energy storage mechanism. And then the organisms use it as a battery of some sort and then produce CO2 to live and the CO2 comes out. So without looking at the system impact of this, so if the microorganisms don't do that, we will have much less CO2 emission, right?
Yes, that's true. Yeah. We would also have
obviously
much
that's not a
less
sustainable system.
Yeah. Yeah, and the problem is if you have the soil is basically a living organism, and it only functions if we have a healthy functioning biome or like microbial community. So basically, if we imagine a soil where microorganisms do not respire, then yes, we will have fewer CO2 emissions, but we probably also wouldn't have any food to feed ourselves. So we would have a very boring situation of life on Earth, I would say.
Yeah, we'll have a very pristine environment, but then we all be dead. That's not necessarily a good thing either.
Yeah, okay, we're really going off on a tangent, but basically then we would also, plants also need the CO2, of course, to make oxygen for us, and so that the Earth will look completely different.
Yeah, so that's the point, right? So this is a sort of a self-sustaining balanced system. What throws it off is when things are a lot of off kilter. And it could happen from a variety of ways. The antibiotics that we are putting into the soil could kill off microorganisms. And the CO2 concentrations in the air through anthropogenic activities could tilt the system. And I understand that. So this reduction process, as you say, could happen not only in oxygen, but also not only in environments that has oxygen, but also anoxic environments where other metals sort of fill in the gap for oxygen, right? So talk a little bit about that, yeah.
Yeah, so there's different, you mentioned the metals, so the metals are one possible respiration pathways. There are others that we're not going to go into today, but there's also dissolved species such as nitrate, sulfate, or even CO2 that the microbes can use. But I'm very interested in these redox active metal species. And the reason why that is with these metal phases, the respiration is not that straightforward for the microorganisms. So if you imagine that you're a microbe in a soil, you oxidized some carbon, and now you have these electrons at the surface of your cell that you need to get rid of. So for us, for example, we take up the oxygen into our cells. The reaction takes place there, CO2 goes out. But if you're a microorganism and you have like a metal next to you, it can be like several times the size of your body. And you cannot basically bring it inside the cell to carry out this reaction. So the microbes need to have some mechanism to basically transfer these electrons from their cells onto the metals. And that can be done either they could touch the metal directly, Some microbes are known to like form long strands of conducting basically pili, they're called, through which they can transfer the electrons, or they can also use very small organic molecules that can diffuse to the mineral and then come back. And so this reaction is more complex than, for example, the reduction of oxygen inside a cell, because you also have the crystal structure. The electron goes onto the crystal, and then the question is, what happens? Where does the electron go? Which atoms does it associate with? You can also have then a change in the crystal structure of the mineral if you start depositing electrons into the mineral. So there's a whole range of interesting questions related to mineralogy that you can then ask.
And it's more complex.
Yeah. Maybe one more comment. In the soil, these minerals don't just serve the function of being electron acceptors for microorganisms. They also have a structural role in the soil. So they're also crucial for the structure of the soil, which then, of course, impacts again where the microorganisms can live, where their food comes through, where water flows through, etc.
And I understand that some microbes prefer anoxic environments too, right? Even though the energy transfer is more complicated, but that's what they specialize in.
Yes, exactly. So there are microbes that can only survive under oxygen, also when oxygen is present, but there are other microbes that can only survive if there is no oxygen, and then there are also a few that can do both. But many of the anaerobic microorganisms that live under anoxic conditions, they're specialized for these types of anoxic pathways, so they cannot survive under oxygen. And that basically happened because I imagine evolutionary, if a microbe is in an anoxic environment, it would try to adapt to survive in that environment. And I imagine I'm not a microbiologist, but I imagine that's how these pathways developed, basically through niche differentiation.
Niche differentiation, and they continue and diversify further. And so things continue to change. But it seems like this has some diagnostic properties, right? So we can take a soil sample, and if you run it through some sort of analyzer, we could sort of say what the condition is of the soil or underneath that, right? In some ways.
Yeah, in some ways. So we are using a method that basically allows us to mimic what the microbes are doing in the environment. So we have an electrochemical cell where we can add our soil sample with our minerals, and then we can basically act as the microbe and start pushing electrons into the system. And then we can directly through, an electron flow is basically a current. So we can monitor the current, and from this current we can determine how fast do electrons go into different mineral materials, to what extent can we reduce them, and then we can also use additional techniques to look at the minerals, for example, if we're interested in how the mineral changes in this process.
So it's almost like it's an electric process. Electrons flowing back and forth, and protons or hydrogen nucleus flowing back and forth. And so suppose we have an environment that we want to change. Could we use electricity to change it back to some better state?
Ooh. Wow. I never thought about that. I mean, there are some applications with microbial fuel cells, for example, where we try to use this capability of the microorganisms to produce this current, to have a current that we can, that we can, energy basically that we can use for something else. I don't know. I don't think if we apply attention to a soil, we could
possible
change it in that way.
Yeah, another thought I was just going through my head is I don't think this is possible either. Um, if there is some sort of electron flow, could we create a biologically driven battery of some sort? Not just battery,
Mm.
but sort of a electricity producing thing.
Yeah,
uh,
yeah. I think these microbial fuel cells that I mentioned are going in
Microbial
the
fuel cells.
direction to what you're thinking. So
Yeah.
there we're basically feeding the microbes substrates, and then we let them transfer electrons to an electrode, and then we harvest these electrons that come in through the electrons to the electrodes as current. But you need quite a massive volume and number of cells to have a decent current that you can use for something. Yeah.
Yeah, and the feed could be sort of waste material,
Yeah.
possibly some
Yes.
waste, right? So it could produce electricity by consuming waste in some ways. That could be quite interesting.
Yeah, I think the problem with there are approaches that look into this. I believe the problem is the scaling up. So you can do it
Scaling,
on a
yeah.
small scale, proof of concept that works, but I haven't heard of any like big facilities that can use this. Yeah.
Yeah, so I want to talk a little bit about sort of the pH versus EH structure. So EH is what you call redox potential, right?
Yes.
And pH is pH. I think people generally know, but it's proton availability, some sort.
Yes, exactly. So basically, you can think of EH and pH as two similar things that describe different conditions in the environment. So the EH is basically defined as the pressure of electrons that you have in a system, and the pH is the pressure of protons. So you have the positive and the negative charge. And pH is relatively well known, I would say, but EH, I think most people have never come across. But it's a property that is just as important for an environmental system as the pH.
And they were generally negatively correlated, right? I would imagine.
Yeah, yes. So if you in environmental sciences, we have this so-called Pourbaix diagram or EH-pH diagram, where you have the EH on one axis and the pH on another axis, and then you can basically, like people did many measurements across very diverse environments, and then you can basically plot the EH and the pH, and yes, you do see this inverse relationship for. And then you can see where, for example, your anoxic sediments would lie compared to your oxic soils or intermediate level soils or sediments. Yep.
And it's looking at diagram here, Meret. So you say here EH, pH, and dissolved AS, V at AS3, and Fe, iron, as a function of incubation time under flooded conditions for a rice paddy soil. So flooded conditions, so before the flood, there was oxygen in the soil. After the flood, there is much less of it. So it sort of changes the whole thing, right? Changes the whole
Yep.
community underneath there.
Yeah,
So
and the
what
chemistry.
happens?
Yeah.
Yeah, what happens in the flood? Yeah.
So, but yeah, if you have a soil that is well aerated, you're going to have a certain like community of microorganisms that are well adapted to breathing with oxygen. Then once this oxygen is used up, so that can happen, for example, under flooded conditions, where the microorganisms consume the oxygen faster than it comes in through diffusion. And then what the microbes do, basically their most energy efficient respiration pathway is now no longer available. So they switch to alternative respiration pathways. And what they would first use is nitrate, because that produces the second most amount of energy.
Yeah.
So then the community that can use nitrate has a happy life until the nitrate is used up. Then they're going to move to the next electron acceptor, which would be minerals containing manganese or iron. But there, the span over, or the conditions over which they can use them is really not as well defined as for nitrate, because the properties of these minerals change quite dramatically depending on which mineral we have, what the solution conditions are, what, for example, the size of the crystals is, and other factors. So we don't have as good an understanding of these processes or of these materials. And then you can move further. If the minerals are all reduced, then you can have sulfate reduction and methanogenesis eventually. But it's also important to know that, like, this is what we refer to in environmental chemistry as the classic redox ladder. But in an environmental system, especially in soil, you can have conditions that are so heterogeneous that you can have one process next to another. So that's also something that we investigated in one of our papers. What can happen in a soil is that the structure is very heterogeneous. You can have different aggregates, you have flow paths of water somewhere, so you presumably have very different conditions for the microorganisms because you have different availability of the substrate they eat, you have different availability of the electron acceptors they use. So what can happen is, in a soil, you can have a pocket where microorganisms have a lot of carbon and they eat it really fast. So they consume all the oxygen faster than the oxygen would diffuse into that pocket. So then you could have anaerobic processes in that pocket, whereas like a few centimeters or millimeters or meters, depending on the soil away, you could still have oxic processes and you would know nothing about these anoxic conditions, basically. And that's a big uncertainty when we try to model, for example, the carbon cycle. We don't really have a good understanding of these anoxic pockets. And so, if we don't know how much of the soil is anoxic, we cannot really accurately predict what greenhouse gases and how much of that we would expect to be released, how fast.
So when these microbes switch these energy channels from oxygen to manganese to iron to others, are these the same microbes or are they different microbes?
They're different microbes.
Different microbes. And so the ones that were using oxygen after the flood, they're going to get wiped out. And so some other species is going to thrive and dominate. So when the flood recedes, what we have is a different, just like the human microbiome, we have a different soil microbiome. And then it takes time, right, for it to sort of come back because all the good guys, so to speak, who are using oxygen are gone. And they have to come back and start doing that again. So you said what we find. So when a land is flooded, tactically, let's say the flood lasted only for a few months or something, and the flood goes. That land's characteristic has substantially changed from a microbiome or microbial perspective. How long does it take for it to normalize again? It depends, I guess, but
yes, but not super long because the generation times of microbes are relatively short. You can also have certain microbes can go dormant, so they're still there, but they're not active, but they can grow again quickly when the conditions are favorable. So we actually, you mentioned these flooded soils. We here in the Swiss Alps, we basically see this each year because in winter we have snow cover. The snow melts, like right now it's starting to melt, so the soils will get flooded, but then they dry up again over summer, and then the next winter comes, basically. So we see this annual cycle also, and we see the shift in the microbiome with these different seasons. So yes, they do recover, but you do see interesting changes with the different redox conditions, definitely, yeah.
So you say because they're dormant in some ways, so they come back fairly quickly. So, in human diseases, we have ways to sort of change your microbiome to treat some diseases, but this is not needed, it sounds to me, for the soil, because stuff is still there, but they're just going to switch back and forth between channels.
Yeah. Now I'm going out on a limb a little bit because I'm not a microbiologist. I'm a soil chemist. So we look at what the microbes do to the chemistry. So my understanding of the microbes is okay, but I'm not an expert. So I know that certain microbes, at least in the Swiss Alps, they go dormant over winter, then they come back. I don't know whether that's a general trait of microorganisms or whether these are specialized microorganisms in this type of environment.
Yeah, that's interesting. So when things are expected to cycle, like in the Swiss Alps, there has to be some sort of a mechanism that says, the winter is coming, I'm going underground or wherever, and then I stay there for a few months, and then I'll come back up again. So just like any other animal, I think they are hibernating.
Yeah, in a way, yeah. Yes. So it's basically a temperature effect. Once the temperature and also availability of organic carbon or food, because the plants, of course, also stop producing or stop photosynthesizing towards the fall. Yeah.
So I want to go into some of your papers, Meret. So the first one I have is soil carbon concentration drives anoxic microsites across horizons, textures, and aggregate position in a California grassland. You say here, anoxic microsites are zones of oxygen depletion in otherwise well-aerated soils serve as prominent controls for several biogeochemical cycles, carbon, nitrogen, iron, and so on. So zones of hydrogen depletion, anoxic microsites. So could you talk a little bit about that?
Yes, of course. I tried to link to it before already. So basically you can form anoxic conditions if you are in a soil microsite, we call it, or pocket, or just like a smaller area of the soil, smaller than the total soil you're looking at. And in this pocket, you can have microorganisms that are really active and that are consuming the oxygen pretty quickly. And because oxygen diffusion in water is pretty slow, it can be that the oxygen diffusion into that pocket is slower than the consumption by the microorganisms. So then what naturally happens is that the concentration of oxygen in that pocket goes down. And as the concentration goes down, the microorganisms have to switch to these alternative respiration pathways that we talked about before. And that, of course, means that we can have quite drastic changes in the chemistry. For example, if we get iron reduction in that pocket, we can reductively dissolve the minerals and form iron 2, which is the reduced form of iron, which is much more mobile than the oxidized form, the iron 3, which is usually present in mineral form. So you could have, for example, also transport of that iron away from that pocket, or this formed iron could cause recrystallization of the mineral or other processes. But yeah, these anoxic microsites are basically sites where microorganisms consume oxygen faster than the influx of oxygen is.
And in some situations, that could be a problem. So you say here, across the entire data set, our combined proxies for anoxic microsite prevalence were strongly and positively correlated with organic C concentration, highlighting the importance of soil organic C availability and microbial oxygen demand in creating anoxic microsites. So this anoxic microsites, do they have a different color? I'm thinking here, without knowing anything about it. We could take a picture of a cross section, and then maybe use AI or something to sort of measure how bad the anoxic conditions are potentially.
Yeah, that's a really good idea. And actually, yes, we can do it to some degree. For example, if we have iron in that microsite, I mentioned before the iron 3 and the iron 2. So the iron 3 is usually present as minerals of different colors of orange, yellow, brown. But the iron 2 is more grayish. So if you have really a lot of iron 2, you would see, for example, a microsite as a more gray area. And there's these sites are really difficult to like characterize. So one approach is to use synchrotron radiation to look at the redox state of iron. So so my colleague Vincent Noël, who's also on that paper, he's an expert in that. So what they basically do is they scan the entire area, determine where they have oxidized and reduced iron. They can also do it with different elements like sulfur. And then they can say, in the areas where we have the reduced species of the element, we have anoxic conditions, and that's like the extent of our microsites. Now, these microsites are really hard to capture because they're not static. So it's basically a microbially driven process. So these can come and go depending on the temperature, soil moisture. For example, they can have very different dimensions. Like you could have a microsite on a micrometer scale, or it could be centimeters or meters wide.
So if these are agricultural lands, for instance, it will reduce the productivity, I would imagine, right?
Not necessarily.
Not necessarily. Okay.
Yeah, because in these microsites, you also accumulate organic carbon. So you can imagine if the microorganisms, these anoxic pathways or anaerobic respiration pathways are much slower than the oxic respiration or the aerobic respiration. And that's because they basically gain less energy from oxidizing these compounds. So if a site goes anoxic, the production rate of CO2 will drop, or the activity of the microorganisms will drop because they have to basically do more in order to obtain that energy. So what happens typically in these microsites is that we have organic carbon that is degraded so slowly that it persists over a longer period of time than if you compare it, for example, to an oxic soil. Now, if we try to bring this back to agriculture, we know that organic matter is a really important component of the soil to keep the soil structure. So it's basically the glue that holds the soil together. And plants really need a healthy soil structure in order to thrive. So we cannot say these microsites are bad.
So it could be good potentially. So I'm thinking it's some sort of intervention. So I'm thinking from a sort of policy perspective, agricultural lands. We go in and we look at concentration of these microsites. And that may be a good sign. If you don't see, I'm just making this up, Meret, I have no clue. And if you don't see, sort of average prevalence of microsites, that might be a problem. So you could potentially intervene in those lands. So do we use this data to improve the productivity? We talked about paddy fields. I haven't spent much time in it. I grew up in South India, so I have seen them quite a bit. And productivity is a big, as you know, developing world. Productivity of the agricultural land is a huge issue. So there might be information here that we could intervene to improve productivity.
Yeah. So I think you're already too far ahead in your thinking. There's not even that much information on these microsites yet. So there's still several groups actively working on that. As I said, it's very difficult to characterize them. So we're basically a bit stuck in the method development. But to get back to your point of the agricultural land. So we have a project where we're looking at how the tillage of the soils, or the plowing basically of the soil, affects these microsites. And we know that plowing, if you regularly plow a soil, you destroy the soil structure each time. It's known that you can lose a significant amount of the soil's organic carbon, and then you basically end up with a not very fertile soil after a while. So there's a trend to go towards low tillage or no tillage soil. And we have a site here in Switzerland where they had different tillage practices for decades. So really like long-term sites. And we looked at how the anoxic microsites, or we are looking at how the anoxic microsite prevalence differs between these different treatments. And so far, it looks like, well, we haven't analyzed all the data, but it looks like the no-tillage soils have much better soil structure, and they also have higher prevalence of these anoxic microsites and tend to have higher organic matter contents.
It's really fascinating. So we have this carbon cycle. So we have plants sort of pumping carbon back into the soil, and we have this microbes using it at different levels. And so it's some sort of an optimization problem, right? So going back to that, I mean, in the forest, things are like how they are. So there are too many management issues. But in agricultural land, this is truly an optimization problem, isn't it? I mean, there are multiple things going on here.
Yeah. Yes, you're right. You also cannot like dump an infinite amount of organic matter into the soil because the microbes are just going to respire it. So that's a bit. There are several initiatives that want to increase the organic or the uptake of CO2 basically into soils. The problem is always that, like, which time scale do you look at? Because if you add, for example, manure or organic fertilizer from compost, it will go into the soil when you first add it. But then the question is how much of that comes back out because the microorganisms just eat it and produce greenhouse gases. And we still don't fully understand what determines whether an organic molecule that comes from a plant, whether it remains in the soil and is stabilized, or is eaten by microbes and returned to the atmosphere. So we know that one very important process is the association of these organic molecules with minerals. So basically, some compounds can stick to mineral surfaces, and then the microbes have a harder time to get it. And so basically, that's what can stabilize these compounds over longer periods of time. But this is still an area of active investigation, and we don't fully understand the processes, and we're also quite a little bit away still from being able to predict what will happen in a warmer climate. Which will, of course, also depend on where on the world you are, what the properties of your soils are, etc.
Yeah, and the history of the soil in some ways. So carbon sequestration was suggested as a way to get rid of CO2 from the air, but that's not necessarily. So you might put carbon in there, and depending on the soil characteristics, it could come out pretty quickly if there is a ton of microbes in there.
Yeah, I mean, there are studies that use the 14C carbon dating method to see how old the carbon is that is in the soil. And you really have a range from like basically yesterday, which you cannot measure with this method, I guess, to like thousands of years. And so like humans, we don't really need a solution for thousands of years at the moment, but at least for a few decades in order to be an efficient measure to mitigate climate change. But yeah.
So if carbon sequestration, I don't think the energy equation works here, but if it does work, it has to be at sufficient depth, I would think, right, for it to be stable down there.
Yeah, it's not. I don't think it's primarily a question of depth, but of the like geochemical and environmental conditions that you have. So if it's unfavorable for microorganisms to eat the carbon, that could be because there's not enough soil moisture, or the temperature is too low, or the organics are bound to minerals, or you're in an anoxic environment where the thermodynamics are basically not favorable. Like these are all things that could contribute to the stability. Like usually if you go further down in a soil, you also lose a lot of the microorganisms, or you have fewer microorganisms usually at depth.
Yeah.
That's also and your carbon concentration generally decreases as well because the carbon comes in from the top. Then and then as the like soils form, it becomes less towards the bedrock, basically.
Yeah, so I want to go into another paper. Thermodynamic controls on rates of iron oxide reduction by extracellular electron shuttles. You say here anaerobic microbial respiration in suboxic and anoxic environments often involves particulate ferric ion oxides as terminal electron acceptors. To ensure efficient respiration, a widespread strategy among ion-reducing microorganisms is the use of extracellular electron shuttles that transfer two electrons from the microbial cell to the iron oxide surface. So as we discussed, so this is after it's an anaerobic situations. There is no oxygen going further down from manganese, if I understand this correctly. So iron is sort of the next rung on the ladder, right? So that's where we are. And you say so. If to ensure efficient respiration, extracellular electron shuttles. So could you explain that a little bit?
Yeah, of course. So these microorganisms, they sit somewhere in a soil or in a sediment. They oxidize the carbon in their cells, but now they need to be able to put the electrons somewhere. And the minerals, so the ferric iron oxides in like complex language, they're outside their cells. So the minerals can even be like super large constructs, bigger than the cells itself. So they need to find a way to transfer these electrons from the cell surface to these minerals. And one way to do this is to use so-called electron shuttles. So extracellular just means they're outside the cell. And electron shuttles means that they can go back and forth, basically between the cell and the mineral. And so many of these shuttles, they can basically take up two electrons at a time. So they would diffuse the microbial cell, take up two electrons, and then diffuse all the way to the iron and dump these two electrons basically onto the mineral. And so in this work, what we did is we mimicked this dumping of electrons from these shuttles onto the iron. And what wasn't clear before is how that relates to the energetics of the system. So people have seen that you can use, they use different types of shuttles with different chemistries. They tried this at different pH, but they didn't find a unifying description for the rate of this transfer as a function of the system thermodynamics. So what we did here is we basically were able to develop these rate-free energy relationships, and that might also be jargon for some of your listeners. So a rate-free energy relationship is a concept that is often used in chemistry that relates the kinetics of a reaction to the thermodynamics. And so based on like general like thermodynamic concepts, you would expect that you have a high rate if you have a high energy release and a low rate if the energy release is low. And if you have these relationships for certain types of reaction, that allows you to basically predict a reaction rate in another system where you know or where you can estimate the thermodynamic conditions. So if we take the iron reduction as an example, what we did in this work could potentially help us to predict or estimate at least how fast a microbe could use an iron mineral as an electron donor in a soil under certain pH conditions.
And so under significance, you say here, under anoxic conditions, various microorganisms couple the oxidation of organic carbon to the reduction of solid ferric iron oxide phases using extracellular electron shuttles. So you talked about that. Determining the contribution of this widespread terminal electron accepting process to total anaerobic respiration has proven challenging because of large variations in observed ferric ion reduction rates. Yeah, so you talked about that too. It almost sounds like to me, Mert, that they say that we know about space a lot more than what we know about the depth of the oceans. And it seems to me that we know a lot more about space than 6 feet under our feet, because there is so much going on there, which has such such significance and such importance to climate control, to energy production, to the health of the planet. But we are just sort of getting into it now. It seems to me, yeah.
Yeah, it's maybe not. Yeah, I think the soil has a bit of bad image because some people, you could just consider it dirt that is annoying and sticks to your shoes, but there's actually much more going on, and it's very crucial to life. I mean, we wouldn't even be here if we didn't have the soil to produce food to live.
And it's so non-linear because of the various things. I mean, there's physics involved, there's chemistry involved, there is biology involved. So it is so non-linear.
Yeah,
that
which is challenging, but also the beauty of this subject, because you get to work with a lot of like other disciplines and interesting people.
Yeah, so this is really at the intersection of disciplines, and I think this is probably where we lacked sort of a thrust into research in this area, because it's not just a soil question. It's a science question, and it's so non-linear. It's very difficult to fully flesh out. So I want to finish up with another paper that you have. So redox properties of solid phase electron acceptors affect anaerobic microbial respiration under oxygen limited conditions in floodplain soils. So we talked a little bit about that. So mountain floodplain soils often show spatiotemporal variations in redox conditions that arise due to changing hydrology and resulting biochemistry. So this is a bit like Swiss Alps, I would think,
exactly.
right?
This was in the
Yeah.
US and Colorado, but it's basically a very similar system where we have a snow-dominated system in the winter, then we have snow melt in the spring and early summer where we flood the soil, and then we have a drying up again and snowfall again the next winter.
Yeah. So if I were to get data on sort of cyclical processes, as you say, in Swiss Alps or Colorado or someplace like that, as opposed to some sort of unexpected flooding, will we see a huge difference in recovery and damage?
I would say yes, because these systems where we have these annual seasonal cycles, they have adapted to these processes. That's also why we have these floodplain soils. So usually the river floods the same plain each year over many, many years. So we formed this floodplain basically. But if you like think about, for example, an extreme like rainfall event where you have flooding of a soil that usually doesn't get flooded, there's much higher chance that you have like damage or significant erosion, or the microbiome wouldn't recover as fast, or you have damage to vegetation.
Yeah. So I know that you don't do research in this area. So from a policy perspective, so based on what we understand now, there's going to be a lot more research that needs to be done. Agricultural productivity, more important for the developing countries, possibly. And we have mechanization of agriculture. So we talked a little bit about tilling as a possible negative effect on the land. So two questions. One is the mechanization going to create a problem in the long run?
I think for soil health, probably yes. But in this case, I think it's kind of it kind of competes with the agricultural productivity. Because if you imagine also with tillage, as you mentioned, I mean, tillage also has benefits because you're able to like till the organics into the soil that would then like benefit plant productivity. So it's not a unidirectional problem. So with mechanization, you mean the use of big agricultural instruments? Yeah. I mean, what we know is that if you have very heavy machinery, you also compact the soil. Roots are not able to grow as well. Yeah, one really big problem, or like the major problem, is that once a soil is degraded, we're not going to get it back within our lifetime or our children's lifetime, because a soil needs thousands of years to develop. So once a soil is really infertile and cannot be used, we basically lost this resource.
Yeah, I would love to see some research in sort of soil treatment interventions. Meaning, so I have some bad stuff happen here. I want to bring it back. And how do I do that? There are multiple ways I could possibly approach it. So there could be interventions that we could deploy, right, to bring soil back.
Yeah. There's, I think there's approaches to basically inoculate soil with microorganisms again, whereby you transfer a functioning microbiome. You try to transfer it into the soil with some like carbon additives so that they have things to eat. Then you can try to plant plants that have beneficial effects and bring carbon back into the soil so the structure can form again. I think it really depends on the level of degradation.
Yeah.
But you need to bring this living organism back to life, and a lot of factors need to be right in order to do that. But it's worth a try.
Yeah, it's a fascinating area. It's fascinating research, Meret. Yeah. I didn't know anything about it. And there's a lot here. And it's, as you say, it's an interdisciplinary arena, because a lot of different areas get into this. But it has long-term implications, I think, for society and, generally for policy. So cool. I hope you succeed in your research.
Thank you very much, Gill. And thanks for having me on. It was a pleasure talking to you.
Yeah, thanks for spending time with me.